Hi guys, it's Miss Davenport again, and we are continuing with the cell transport topic. This is just part 2 because I didn't want you to sit through like a 15-minute video. I wanted to be able to break it up. So we do have practice problems. These are more likely the types of questions you're going to see on tests and quizzes than simply what is active transport or what is passive transport. You're more likely to see these types of questions. So 3 things to always keep in mind about cell transport problems is water moves from a high to a low concentration. All of these practice problems are talking about osmosis. They're talking about the movement of water. We're not focused on where the salt or where the sugar or anything else is going. We're focused on where that water is going because we want to know if the cell is going to grow or shrink as a result. The plasma membrane is semi-permeable for a reason. That means it's not going to let everything through. It's not always an open door welcoming anybody and everybody in there, right? At clubs, they don't want to let just anybody in, okay? They're selective. Okay, so semi-permeable, another word for that is selective. The plasma membrane only lets certain things in and certain things out. If your membrane was just open all the time, think of how often we would be sick. Think of how often things would just go wrong and life would be crazier than it already is. Okay, and water follows salt. Salt draws the water out of things. Things want to be balanced. So if you have a really salty environment, it wants that to be more diluted or balanced. So the water's going to follow that salt wherever it is, whether it's inside the cell or out of the cell. When you want to dilute something, when you want to balance it out, you add water to it. Think about your Kool-Aid, your lemonade, your coffee, whatever. You're going to add water to it to make it less strong. We all know if you pour salt on a slug or another organism that's very moist, that salt, you can literally see that slug kind of shrivel up because that salt's pulling that water out. The slug's environment internally wants its outside environment to be equal as well, so the water's going to leave its cells to try to dilute the salty area. Of course, not really realizing that that's going to ultimately lead to its death and that homeostasis and balance cannot be reached in that way. But water follows salt. Salt draws things out of your cells. That's why they also tell you to gargle salt water when you're sick. It pulls the fluid out of your cells. They burst, so that makes swelling go down. It also kills any bacteria or viruses that might be in your cells as well. So, on to the problems. So you'll see something like this. Okay, giving the following internal and external conditions, what would happen to the following cell? So we have to look at the different parts we're given. This is the inside of the cell. The concentration is 80% water. On the outside of the cell, it's 20% water. So we have to think to ourselves, where is there more, where is there less? We got to find the high, we got to find the low, because water goes from a high to a low concentration, so it can balance things out. So in this situation, this is our high concentration right here inside of the cell. Outside of the cell is our low concentration. Water goes from high to low. So we're going to draw our arrow going out, which means the cell's going to shrink. This water is going to come out here, try to balance this. So it's like 50-50. So 30% of that's going to go in there to make it 50-50, to make it balanced, to make it equal. And that's going to make that cell shrink. High to low. If it went this way, then we'd just be dumping more water into the cell and the cell would grow, and then the water on the outside of the cell would not be equal at all. Okay, it's about keeping things equal. Let's try some more. Right, this time we're given NaCl. That's another word for salt or sodium chloride. Sometimes you're given it like this. You're not given it so easy where it gives you the water concentrations. There's 2 ways you can do this. If you wanted to, you could find out the exact concentration of water. You could subtract each of these things from 100, see how much water you have left over, see where the high one, where the low one is, and where it's going. But remember, I gave you a little bit of a cheat. Water follows salt. This water is going to go where there's more salt because it wants to dilute things. So where do we have more salt? We have 5% salt on the inside of the cell and 0.5 on the outside. There's more salt inside the cell. The water's going to follow that. So the water is going to go inside of the cell to try to dilute this out, make it less salty. Where there's more salt, there's less water. So the water's got to go in to balance that out. We're going to try some more practice problems. So if you're still a little bit confused, stay with me. Okay, so extra problems. Okay, this one, pretty easy. You're given the water concentration, 50 on the inside, 50 on the outside. Trick question, is anything going to happen to this cell? No, it's already equal. It's already at homeostasis. It's already balanced. Nothing needs to happen. So the cell is going to stay the same. What about this one? Okay, think about it for a second. Where's the high concentration? Where's the low concentration? Water goes from high to low. Is it going to go inside the cell? Outside of the cell? We know it's not going to stay the same. Here is our high concentration outside of the cell. Low inside, high to low, the cell's going to grow. Water's moving inside to make it balanced. Let's try some more. Ooh, here we go. We got that salt again. Don't let it throw you off. Where's the water going to go? We have 0.1 inside, 1 outside. 1 is bigger than 0.1. There's more salt out here. Water wants to dilute it. So water's going to follow that salt, and the cell's going to shrink. We got one more for you. Oh, actually we don't. That was the last one. If you want any more examples, just feel free to ask any of the biology teachers during class, and we're going to be working on this so that you guys get it, because it just takes practice. It's almost kind of like math in a way, where the more you do it, the more you get it. So hopefully you learned something and you're not completely lost and completely confused. And thanks for watching.